tired, it's the end of the semester. Have you seen that dog that's like barely struggling to stay above water? It's like all of us right now. Just trying to keep breathing. Okay, you guys are not alone in that. All right. So how many people have actually looked at the data analysis stuff? Oh, more than no one, that's good. Just for reference, can I get a show of hands of people who plan to come to lab at some point tomorrow? Okay, it's good. Again, fine, we don't. Okay, all right, so, yeah, I'll be here at 8 and we'll just plan to sort of meet in the lab. People can come and go. Depending on how many people we have, we may actually move to the research space for a little while, which is actually good because then you can see how the 310 runs. It'll be interesting. But that just sort of depends on who's here and how many and what we're doing. So again, tomorrow in lab, if you're going to show up, we'll probably just, I'll let you go through the data analysis piece and I'll be there to help you figure out what the heck is going on, show you what you need to know. I can go through other lab report stuff. Next week, we have Tuesday and Thursday labs, which will again be open. Those I'm going to run more as review sessions, unless somebody asks me to do something different. So on Tuesday, we will review new material. So we'll have additional problems or questions, or we can go through the worksheet. Wednesday will be new material exam. Thursday, we'll have open lab for cumulative review. So we can review any concepts from any point this semester. And then we'll have our cumulative final. So the cumulative final will be all multiple choice. There will be no long questions, I don't think. I have to go back and check sort of the format. I started writing it last week, and I've already forgotten what I wrote, so I will go back and check on that. I will let you know if it's different. Yes?
Is the final like a certain percentage, or is it just like another test?
So the combined two of them is 15% of your grade. A normal unit exam is 10%. So they will probably be pretty evenly split. Like Wednesdays will be about 7.5% and Fridays will be about 7.5%. But they'll go together to 15% then. Yes?
With the cumulative one, are we taking the same test that we took in the beginning, or
Sure
different?
are.
Okay.
Yes. So that pre-test column that everybody's deeply stung by right now will, remember that grade will be replaced as long as you do better. I will again tell you, I've never had anybody do worse. I have had some people who have stayed consistent, but I expect that most people will do significantly better on the final, so that will replace your test grade. Yes. Yes, I will have old tests available in lab times next Tuesday and Thursday. It's a good chance to come and look at those and to let me know the mistakes I made while grading them. So please do that. Yeah. Other questions? Don't forget there is an extra credit opportunity. Those of you who want to bolster your grade a little bit and do some extra studying, good chance there. All right. This is the first time I've been able to see everybody clearly in 2 weeks. It's amazing. I know. Apparently, like, went blind in my left eye, and my glasses are super old, so it's awesome. I don't have a headache. Ready to go. Okay. So we are going to come back to our allele and genotype frequencies and extend this idea into Hardy-Weinberg, which I know some of you get introductions to Hardy-Weinberg in Bio 101, 102, and or Ecology and Evolution, depending on who you've had and what you're doing in there. And to some of you, this may be new, so we're going to take it from the basics, which is where we started on Monday. All right, so here we calculated the frequency of the G allele, little g allele. We found that that frequency was 0.2 or 20%, is that correct?
Yeah.
Okay. So then we went to calculating genotype frequencies. I will remind you again that it is incredibly important when you're looking at questions on worksheets or in the textbook or on an exam that you really read and pay attention to the terms, right? Because I may ask you for the allelic frequency. Don't confuse that with the genotypic frequency. Those are two different questions, right? So, and I'm not trying to trick you when I ask those questions, but I'm just telling you now that you will need to be aware of sort of word choice. So with genotype frequency, we calculated genotype frequency for little g, and we found what? Did we do this? No. Oh, excellent. Let's do this one. Okay, so our equation for genotype frequency is going to be the number, oh, I gave a different example. Number of individuals with a particular genotype over our total number of individuals in that particular population. Okay? So we are working with our frogs again. We have a total population size of n, and in this case n equals 100 frogs. What just happened? I wasn't even hear it. Okay. So here we have already given to us the number of individuals with each of our genotypes. So if I laid it out like this should be easy for us to calculate, because all we do is we say total number of individuals that are homozygous recessive over 100, which is what? Okay, so 4 over 100 equals 0.04, which is 4% of our population is homozygous recessive. Okay? So, when we're dealing with allelic frequencies and genotypic frequencies in a population, the numbers that we calculate should add up to 1, which equals 100%, which just means that we've accounted for every allele or the genotype of every individual in our account. Okay, so if we go back a slide and we say, right, so this is our frequency of homozygous recessive individuals, 4%. We can also quickly calculate our frequency of heterozygotes, which is what? 32. Okay, 32 over 100 equals 32%. And our frequency of homozygous dominant individuals, 64, thank you, over 100, 0.64 equals 64%. Right, so in this case, what we would, and this is an excellent way to check your math, because if you calculate 3 genotype frequencies and you add these 3 numbers together and you get 0.88, something's not right. Okay. So these 3 numbers should add to 1, or your percentages should add to 100. I think we did okay. Right, and again, that just means that we have actually accounted for every individual in our population. We know the genotypes of every individual. The same thing is actually true if we go back to allelic frequencies. So we calculated our allelic frequency for little g. So our frequency of little g was going to be 2 times 4 plus 32 over 200. I already forgot what that was. 2? 0.2? Yeah. Okay, the other one, we could calculate the frequency of our big G allele. This time we would have to do 64 times 2. Yes? Okay, so 64 times 2, which is 128. Okay, plus 32, thank you. And that should give us 0.8. So again, we can add these 2 numbers together, and that should always equal 1 or 100%. Because again, we've accounted for every allele at this particular locus carried by the entire population. you ever get anything over 1 or under 1, you've either miscounted, you've divided by the wrong number. Okay, so that's what this slide is saying. For any given gene at any given locus, your allelic frequencies should add to 1, and your genotypic frequencies should add to 1. If an allele, or if a locus is monomorphic, so if there's only one allele present, it's automatically 1, because everybody has the same allele. Right? So if we say this particular gene is monomorphic for this allele, what is the allelic frequency? 1. Everybody's got it. 100% of people have that one allele. So there are a couple of ways that we can get to this value of 1. So over here, for our allelic frequencies, I actually did the math for both of them and then made sure they added to 1. If you're really confident that you did the math for one of them correctly, what you can actually do is use 1 minus the allelic frequency of the other to get the allelic frequency of the second one. So right, if we wanted to know the allelic frequency of big G, and we already knew the frequency of little g, we just take our total, which is 1, and subtract the frequency of little g. Right? Either way, you should get what you get. So now we are going to introduce and go through Hardy-Weinberg. So Hardy-Weinberg is an equation that we use to sort of help tell us what is happening in a population in terms of allelic and genotypic frequencies over time. So we're actually, in this case, looking sort of generation to generation. Hardy-Weinberg is a null hypothesis. What is a null hypothesis? The hypothesis of?
The opposite of the hypothesis.
Okay, so it would be the opposite of our alternative hypothesis.
What we accept if it's not significantly different.
Okay, yes. So if it's not significantly different, then what is true about the two things we're comparing? They are, that's another way to say that, the same, right? So our null hypothesis is what we call the hypothesis of no change. Nothing is different between our two populations. Nothing is different between our two time points. Okay, so under Hardy-Weinberg, we call it Hardy-Wine equilibrium. If a population is in Hardy-Weinberg equilibrium, what that means is that no evolutionary force has changed their allelic frequencies from generation to generation. So, in the absence of evolution, we would expect Hardy-Weinberg, or we could sort of use Hardy-Weinberg as a way to predict allele stability. So we're going to go through some examples of Hardy-Weinberg. Hopefully it'll make more sense. If a population is in Hardy-Weinberg equilibrium, that means that allele and genotype frequencies do not change from generation to generation. That's going to be a really important point. So star it, highlight it, put a note by it. Okay. So if we are simply thinking about a gene pool, right? So let's say again that we're a gene pool, and we are in Hardy-Weinberg equilibrium as a gene pool. What would have to happen in order for our next generation to have the same allele frequencies and the same genotype frequencies as what we see in here? Say that again? Parthenogenesis. Interesting. Yeah, I suppose that would do it. All right, assuming we are sexually reproducing species. I was actually just teaching my Bio 100 course about parthenogenesis, and they did not think it was as cool as I do. And I was talking about asexual reproduction and how like budding is an option, and I always, every time I talk about it, I imagine like humans bud, and you just have like a little version of yourself like coming out of your side, and it just like falls off. Budding is crazy. Isn't that weird to think about? That's what plants should be. Yeah. Okay. You digress. I digress. Okay, so what would have to happen in order for the allelic frequencies and genotypic frequencies of a population to stay the same from now into the next generation? Or we could look at it the other way, what might cause them to change? Yeah.
If they mate outside of their group? Or
Okay, so we would call that migration. If we have anybody come into our population and add to the next gene pool, that would change our allele frequencies because they maybe carry different alleles than what we have represented here.
Mutation.
Mutation, right? So we have one of our children mutated. They are now again adding a different allele or different genotype to the population as a whole. What else? How many of our population's individuals would have to reproduce in order for the next generation to look the same as this one?
Every single one?
Everyone. Does that happen?
No.
No. Right? So, like 60% of people end up having offspring. That means that those people contribute to the next gene pool, but the rest of the people do not. Right? So, in order for our allele and genotype frequencies to be the same, next generation, everybody would have to contribute, and everybody would have to contribute in the same number. Okay, so, somebody who had 1 kid contributes less than somebody who has 12 kids. Right? That changes our genotypic frequencies. So there are actually quite a few things that will change the frequencies. I'll give a specific list here in a little bit, but before we do that, I want to show you the equation and start going through examples of it. So what Hardy-Weinberg does is it actually takes our genotype frequencies and it converts them into variables. Okay, so here we have our big G allele. Under Hardy-Weinberg, we would actually call this P. And our little g allele frequency we call Q. Okay, so we're going to talk about P's and Q's. P is always going to be, for us, associated with our dominant allele. Q is always going to be associated with our recessive allele. Again, for our system and for our purposes, we're going to stick to sort of our simplified version where each gene only has 2 allele options. So you're either P or Q or both. But there's not a third variable or a third allele. Okay. So what we already know is if we take the frequency of big G and little g and add them together, we should get 1. That is actually a staple tenet in Hardy-Weinberg. So P plus Q equals 1. Good? All we've done is change the frequency of G and the frequency of Q, G, big G and little g, into P and Q. Add them together, we should still get 1. What Hardy-Weinberg, actually 2 different people, came up with was this equation in the box. So this is Hardy-Weinberg's equation. P squared plus 2pq plus q squared equals 1. P and Q, allele frequencies. P and Q are allele frequencies. Each of the terms in Hardy-Weinberg, so P squared is 1 term, 2pq is the second term, and q squared is the third term. P squared tells us this, frequency of big G. 2pq tells us this, frequency of heterozygotes. Q squared tells us this. Okay? Okay. And again, equals 1 is important because when we add all of our genotypic frequencies together, we should get 1. All right. So, we already have the answers for these, but we're going to work them out using Hardy-Weinberg. In our frog population, where we know that P equals 0.8 and Q equals 0.2, what would the frequency of homozygous dominant be? And sort of the way I'd like you to put this in your notes is the frequency of big G would actually equal, and we're going to put p squared. So we can remind ourselves which terms or which pieces of Hardy-Weinberg go with which calculations. Right? And then, so frequency of big G little g would be 2pq. And frequency of little g would be q squared. And I know that we already have our values over there, but maybe actually do the math using this way so that we can prove to ourselves that this actually does, in fact, equal what we have. And you will have to remember your order of operations again. So exponents first, multiplications second, addings third. So what is 0.8 squared? All right, 0.64. Boom. Any math? 2 times 0.8 times 0.2? 0.32. And 0.04. Okay. So, to take us back a few weeks, does everybody recognize our Punnett square? Yes, good. All right, so we can actually use Punnett squares to sort of verify or to sort of visualize what's going on with Hardy-Weinberg. I'm actually going to throw a little bit of a change in here. So this is actually shown as a Punnett square. In Punnett squares, all of our squares are the same size. Notice how we've always drawn them. We can actually change this into a unit square to better visualize what we're talking about. Because now we're talking about a population instead of an individual. So if we have an individual that is heterozygote, 50% of their gametes will be big G, 50% of their gametes will be little g. Yes? So if we're talking about a population and we're taking all of the gametes that that population can produce and sort of putting them together into a gene pool, 80% of those gametes would carry big G and 20% would carry little g. So the way that I like to visualize this is actually with our unit square, which makes our boxes sort of equivalent in size to the ratio of gametes. So this isn't going to be perfect, but if 80% of our gametes are big G, then we would make approximately 80% of our box represent that big G. And then if 20% is little g, right, so we would do this, which is kind of what's shown up there. Do the same thing on the other side. And so using this information, we can do two things. One is we can, again, figure out, just like we did before, what our offspring will be genotypically. So we will have some homozygous dominant. We have two boxes that become heterozygotes. And then we have one box that is homozygous recessive. So that's the first piece. The second thing that we can do is actually proving Hardy-Weinberg. So the odds of getting a big G from one parent and a big G from another parent. Remember when we did probabilities? So how would we determine the probability of being big G in this population? Yes, thank you. We would do 0.8 times 0.8, guess what that equals? Good, it's also p squared. Okay, so the probability of getting a big G from mom and a little g from dad would be, okay, 0.8 times 0.2, and if we want to show that mathematically, that would be p times q. Yes. Okay. Over here, we'd be getting a big G from the other parent and a little g from the other parent. So again, 0.8 times 0.2. That is also, oops, p times q. Okay. And our last box, which is our littlest box, because it is the smallest value, right? Representatively, 64% of the population, 4% of the population. We would do 0.2 times 0.2, which is 0.04, and equals q squared, right? Q times q. You could also add a p, q. So, since we know this is our population, we can add together all of our different boxes, and that should give us Hardy-Weinberg's equation. So over here, we start with p squared. Oops. We add to that PQ and PQ, which, guess what? There's 2 of them. Good. And then q squared. And that gives us 1 because that gives us everyone in our population. You guys don't seem as impressed as I feel like you should feel.
This is your field.
It should all make sense. Look at all those ways we just talked about allele and genotype frequencies, and our math added up every time. We are on fire.
I hope so, if we use the same numbers.
You would be surprised.
True.
Well, yeah. We'll see on the final. Use all the ways to figure it out. Right? Don't restrict yourself to it. Okay. So if we want to look at a population and sort of figure out the different frequencies of our alleles, this is an excellent chart to use. So we have 3 lines here. The 3 lines show us the frequencies of our different genotypes. So darkest line is homozygote dominant, middle shade line, heterozygotes, light line is homozygote recessive, right? And what we are actually looking at on our x-axis is the frequency of, that's G, little g allele. So if the frequency of our little g allele is close to 0, and we think about Hardy-Weinberg, so if we have something that's close to 0 and we plug it into our q squared, we're going to get very few homozygote recessive individuals. If we also think about Hardy-Weinberg and we say, okay, well, the frequency of big G is going to be 1 minus little g, and little g is very small. That means that our frequency of big G is very high. We get a lot of big G individuals. And then our heterozygotes have this nice sort of bell shape here in the middle. If frequency of one of the alleles is low, then again, we can't have very many heterozygotes, because in order to have heterozygotes, we have to have both alleles present. So at either extreme, we'll have very few heterozygotes. Where on this figure are heterozygotes most common? At what frequency of little g? 0.5. So if our frequency of little g is 0.5, our frequency of big G is 0.5, at those frequencies, we will have the most heterozygotes. If we go either direction, we end up with more homozygote dominant and homozygote recessive. All right, so let's see. Oh, I found an example of this. Let's talk about all these conditions under which Hardy-Weinberg can exist and the conditions under which Hardy-Weinberg cannot exist. So again, what we use Hardy-Weinberg for is to predict stability in allelic and genotypic frequencies from generation to generation. So I'd say here's a parental generation, here are our allele and genotype frequencies. are they going to be stable in the next generation? In order for them to be the same from generation to generation, all of these conditions listed up here must be met. If any of these conditions is untrue, then that population is evolving, most likely, and is not in Hardy-Weinberg. So let's look at these. We have no mutations. We have no genetic drift, which we haven't talked about yet. But that just means that there's sort of random changes in allelic frequencies. We have to have no migration, so we basically have to have a closed population, right? If we open these doors and we let people into our class, that's migration. We immigrate, or if we let people out, that's also migration, right? We lose some genetic variation that people leave. No natural selection, so no selective pressures. Random mating, and large population size. So, would we be a large population size? No. Do we have equal sex ratios? No. Right. Random mating very rarely happens. Okay, so most people do not randomly mate with just, we should take this away from people. Most non-humans, I mean we can talk about humans, but humans get really complicated. But either way, we'll talk about humans. Most people have a preference, right? They don't just mate with everyone that is of reproductive age. Maybe some people, probably not everyone. Right? That immediately violates that principle. The fact that we tend to be monogamous in our social construct deviates from random mating. Yeah. So, are there populations that satisfy all of these criteria? So think about all the species you know. Can you think of any that would meet each of these criteria? In the wild, in the lab?
Bacteria.
Bacteria are asexually reproducing. That changes. You can still do Hardy-Weinberg, but it changes that piece a little bit. Also, bacteria, they mutate pretty darn fast. Yeah. So, no, the answer to this question is no, we don't really have a population or a species that meet these criteria. Simply, and actually I put large population size there. Many textbooks or resources that you use will say infinite population size. Clearly we can't have infinite population sizes. So immediately, there's really no populations that meet these criteria all the time. There are some which are really, really big and are sort of restricted to an area and don't have a lot of selective pressures on them that can get close. We use fruit flies as a model for this sort of thing because we can make really big populations and we can sort of control these other factors in the lab. So most of the time when we do Hardy-Weinberg, what we're actually expecting is that a population is not in Hardy-Weinberg equilibrium. And so there are changes from generation to generation. That's okay. That tells us that something is happening in that population. And so, for example, as a wildlife geneticist, if I see deviations from Hardy-Weinberg, the population is super small, and there's no migration, that helps tell me what's going on within that population. So we can sort of look at these patterns. Okay, so we are going to go through an example. And I know you guys have been really missing chi-square since we didn't do it at all in the last unit. So I'm bringing it back. We're going to do Hardy-Weinberg. I know you were thinking like, okay, Dr. Brandt, how do we actually tell if there's a difference? I know. So here we are. We use chi-square to tell us if there's a difference between our generations. So we are, excuse me. In this case, we are going to consider an Inuit population in Greenland that has about 200 people. They have a very unique blood type. We kind of differentiate, we call it MN and MM blood types. You may remember this from a previous worksheet that you did. Excuse me. In this particular population, these are the breakdowns of the different genotypes. So of those 200 people, 168 of them are MM, 30 of them are MN, and 2 are NN. Okay. So here are all the things we're going to calculate. I think this should be the order that we start in. So the first thing that I would like you to calculate, I'm just going to leave this up here, and you're going to sort of do it on your own, and then we'll come back as a class and talk about it. But start with the frequency of our different alleles, right? So this would be frequency of P and frequency of Q, P and Q. Calculate those. Once you have P and Q, I've sort of outlined it here, expected frequency of each genotype, and then the expected number of individuals. So I realize I gave you exact or observed numbers on the last slide. Here you're calculating your expected values. So how do you calculate an expected value? You take your expected frequency and you multiply it by what? Total number of individuals in your population. Okay, so you can work with a friend, a neighbor, a colleague, and we'll walk around, and then we will come back and we'll make sure everybody has their calculations correct. Okay, we'll come back to that one later. So for these, they'll go back to.
So is this using,
So you go back to this.
oh, this, okay.
Yep. So you're looking at how many alleles do you have total, right? So if you have 200 people, how many alleles do you have?
Okay.
And then how many of those alleles would be M? Maybe your phone's No, no. You're good. Nope. How many alleles do we have? So this is people. Okay. 400. You don't want to, yeah, there you go. You guys, okay? You're doing. Okay. Yep. Okay. Yeah, so your frequency of M would give you your p-value, and your frequency of N would give you your q-value. Good? Got it? Good? Okay. Good? 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 Makes sense? Good. Alright, so I think most people are finishing up, is that fair? Yeah, looks like it. Okay, so let's just run through this math together, make sure it looks like, I mean, I walked around and everybody had the right start, so let's just make sure that we got it all the way through. So how do we calculate our p-value? I know every single one of you had this written down, so somebody tell me.
168
Okay, 2 times 168
plus 30
plus 30.
over 400.
Over 400. Okay, good. This was, again, the most common error was unsureness, uncertainty on what this number should be. Remember that this should be 2 times the number of individuals in the population, which would equal the number of total alleles we have at that locus. Everybody has 2. We're going to assume everybody has normal chromosomal count. So what do we get here? Say it louder.
0.915.
0.915, or 91.5% of the alleles in the population are N. What do we get for Q?
2 times 2 plus 30.
Again, over 400. And that should give you 0.0. Yes. Okay. So from here, for frequency of
Our cells.
it was up there when I went through them a few seconds ago.
Go back.
Alright. Okay, so here we would just sort of plug and play, right? So we know P, 0.915 squared equals what? If we're doing Q squared, we'll just go ahead and go down and do that. 0.085, ooh, 85 squared. Say that again?
0.0072.
Oh, it's back. So, let me say 0. Okay. Oh my gosh. God, I get so flustered. That should be the erase button. It is not. All right. So then here we have 2PQ, right? So 0.915 times 0.085 equals 0.156. Did everybody get to this point? Hopefully with less stress than me. Okay. So from here, what we need to do is calculate our expected. Again, on the previous slide, I gave you observed values. That's what the population actually looks like. Our expected values, we would take this frequency and we would multiply it times 200, which is our total population size. So we'd say 0.837 times 200, and what do we get there? Okay, 167.4. For our homozygous recessives, 0.007 times 200, 1.4, and then down here, 0.156 times 200,
31.2.
So, we don't have time right now to go through our chi-square as a class, so I'm going to give this to you as, do it between now and Friday, and we will go over the answer in class on Friday. You should now be set up. I've given you the equation here at the top. You have your observed values. Those are 2 slides ago, 168, 32, and 2. Expected values were what we just calculated. From there, you should be able to do the rest. Okay. All right, so we will check that out on Friday, and then we will start going into selection. Just as exciting as today. I will see some some of you tomorrow. You don't have to come right at 8, whatever you, anytime between 8 and 10:15. I'll be here. Yeah. Open labs.
Do you know when you're gonna share the policy?
Yeah, now. No, I have a meeting now. Next time you see me, now.
I'll be sitting there for 4 hours, so your bud's gonna like look at me and be like, oh yeah, that's a thing.
I have a meeting, class, meeting.
Gross. Just cancel all of them.
Take a break.
All of them hide under your desk.
Like, people get real into it. So that's what we do. You guys should just sign up. So I don't know because they are being classes from my desk. Are they? Yeah, a lot of them. So they're maybe not letting.
Mommy, don't worry.
Yeah. It's not an urgent rush, right? Because you actually have until the first 2 weeks of next semester to get signed up. But I just want to. And you're old January, like, okay. Yeah. Yeah, so just, I mean, I would prefer if you signed up before. Oh, me too. Yeah, me too. No, I think like even like, yeah, in January, like. Sure. I want there to be no question. Yeah, absolutely. Yeah, for sure. So like, I guess I could wait, hopefully like.
He's got to realize, you got to
Yeah.
you got to
They
pilot
are
license.
usually the registrar's office is usually about like.
Right? Yeah, I don't even know, dude. The game we won at MSOE, we showed up like an hour before, we warmed up for like 45 minutes and played. It was good. We didn't warm up for like an hour and a half, but
Sitting around.
yeah, whatever. That's
Good
good. So
luck.
yeah, thanks man. Have a good one.
How's it going? Everyone's at their max right now, I think.
I'm above the max.
I understand that feeling too. Less than 2 weeks, right?